Hello and welcome to Biology Crash Course Form 4 Chapter 4. Now, the first thing we are talking about the chemical compositions, which are the carbohydrates. Carbohydrates are the primary source of energy in cell. Then you got the lipid, which is the storage of energy. Then you form adipose tissue, which basically just means fat tissue. Now, fat tissue has two functions, which acts as a physical protection and heat insulator, and it can also be used to provide energy when your body requires it and the carbohydrate supply is not enough. Then afterward, moving on, we have the protein, which actually is used for building new cell, renew damaged tissue, and synthesis of enzyme. Now the fourth one is going to be nucleic acid, where they are DNA to store the genetic material, and RNA is used when synthesizing protein. We will learn this more in Form 5 once again. And it's like a basic unit. The basic unit of all this DNA is actually three of them. You have to remember SPM comes up very often. So it's going to be phosphate group, nitrogenous base, and pentose sugar. Pentose sugar is the one that looks like a pentagon, while phosphate, as the O stated, is a round one, and the nitrogenous base is the square one. Now moving on, we have the water, where it is used for maintaining the body temperature. It's a universal solvent, and then it's actually helping for lubrication, support, and transporting a medium. Now, let's talk about carbohydrate here. Carbohydrate, there are three kinds of carbohydrate here. When you just form a carbohydrate, it's only like one slide here, it's like monosaccharides. So monosaccharide is like the most basic form of carbohydrate. It provides the energy. So a few examples of monosaccharide would be the glucose, fructose, and galactose. Fructose is found in fruit, while galactose is found in milk. Now, they are all reducing sugar, so basically as long as you use Benedict test, there's no need to add hydrochloric acid or other stuff in it. Just do Benedict test, and it will work. It will detect it. Brick red precipitate will be formed. Now disaccharide is going to be after you have combined a few together. So when you combine glucose and glucose together, they are like two together. That's why we call it di. Di stands for two. Glucose and glucose together is basically going to get maltose. And if you add glucose and galactose, you get lactose, which is still milk, because we have mentioned that galactose is actually a milk. Yeah, it's a milk sugar. And then afterward, you have your fructose, which is formed, sucrose, which is formed by combining glucose and fructose. So the only thing that's special with these three, maltose, lactose, and sucrose here, is sucrose is a non-reducing sugar. So you can only do Benedict test on it and get result if you have put hydrochloric acid into it. Now, one thing you have to remember is about condensation and hydrolysis. You see, when you add glucose plus another monosaccharide sugar, and the process of adding glucose combining with another monosaccharide is called condensation. The reason is condensation is like water is being evaporating, isn't it? So when water is being evaporating, there are like some substance removed from it, and because of that they have formed bonds between them. It's like, maybe you can say this way, H2O, maybe one of the H is used to combine with the bonds, is used to help to form and link the bond, and thus we form the disaccharide and water. Water is the byproduct of it. Now if the other way around is also possible, where you get the disaccharide, for example, you can get maltose and lactose, then you add water to it, and because the water is present there, the hydrogen bond just decided to go back to the water itself. So that's when we say it's hydrolysis, and you form back your two monosaccharide sugar. So once again, I repeat, condensation is when you are forming bond and the monosaccharides are being formed into disaccharide, while hydrolysis is when you are removing bond, where you put in water and the bond is absorbed to the water, and is hydrolysis. Now polysaccharide is kind of like a lot of disaccharide combined together to form one very long chain. So it is going to be starch, where it is like the reserve in plant in a way, or you can also have glycogen, which is mostly the reserve in animal. We will learn about this when we talk about how glucose is being converted to glycogen most of the time, just to like conserve and maintain our sugar level in Form 5 Chapter 3. And cellulose is going to also be one of the sugar, where it is just meant for cell wall. So once again, three examples: polysaccharide, starch, glycogen, and cellulose.